Thank you very much. So your patient inconvenience. So my name is Minoru Nakayama. So co-author Wioletta Nowak is also joining this session. So our presenting paper is detecting symptom of dementia elderly persons using future of Pupil light reflex. We want to develop a diagnosis for Alzheimer's disease. So there are two approaches in the medical diagnostics. The first one is pathological, to measuring the amount of amyloid beta in the brain. But its procedure is not easy. So usually it's preferred as a clinical approach. It is based on face-to-face consultation. During a consultation, mini mental state or examination, MMSE is usually preferred. So this is consultation marking based on question and answer. This is a scoring. And 23 point, less than 23 point, the participant is maybe Alzheimer's disease. And other scales exist, but usually the MMSE is preferred. So using score of MMSE, 3-class classification is possible. So first one is AD, Alzheimer's disease, MCI, mild cognitive impairment, and normal control. The other factors also influence because most patients are elderly people. Some people taking drugs, something. So then the influenced, and also it's a very important point. Is this procedure required normal verbal communication. If patient have ear impairment, not easy to conduct this examination. So then anyway, appropriate assistance based on any biological measure is required. So first is biological information for Alzheimer's disease diagnosis. So one is oculomotor indices, including pupil responses as biomarkers, and another is conversation voice is analyzed acoustic or lexical feature, and gait analysis is walking behavior is also used for the biomarkers. So we focus on the pupil light reflex as detecting Alzheimer's disease. So because AD influence on retinal ganglion cells connections, and Alzheimer was Parkinson's disease, and sometimes this disease influence to the connection between the retinal ganglion cell to the other parts, transition process. So conventionally, pupil light reflex is compared. And recently, around 2000, IPRGC, it's a kind of a new type of retinal ganglion cells discovered. This ganglion cells respond to the blue light. So conventional retinal ganglion cells, when the light flash happened, once shrinked and move to the distortion process. However, when users blue light and IPRGC respond, restriction is continued to during light flash, and then the response type is a little bit different in the ganglion cells, and also usually pupil response is synchronized the both eye. However, if disease influence to the nerve connections from a retina to EW nucleus, pupil response may be asynchronous. So then we look at the response waveform and response behavior. It may be key to detect Alzheimer's disease. So the purpose of this presentation is we look at wavelengths, PLR's reaction on blue or red light, and left or right flash influence to the retinal connections. The following topic will be addressed in my presentation. The future of PLRs for blue and red light pulse on the left or right eyes or compared. The ability of classifying participant as AD, MCI, or NC using MMSE score and PLR features. And most participant is elderly people. So then the contribution of the participant age will be another topic. So prediction performance of a participant with AD, MCI procedures and developed and evaluated. So we conduct a clinical experiment at the hospital. So clinical survey was conducted. The participant, 101 participants, and the mean age of 78, and so 2/3 is female participants. And this procedure was ethically approved at the Osaka Kawasaki Rehabilitation University Committee. So cognitive function group was classified using MMSE score. So AD is 31. So MCI is only 9, and NC is 61. And how to say 2 of that is normal control. However, normal control includes non-tested participants, and and also age distributions. We classified 4 groups. Is under 65 and 66 to 75 and 76 to 85 over 85. So PLR measurement protocols here. So wavelengths and the difference between the both eyes on lights are compared. So and also pupil oscillation without light pulse were observed. The light pulse is only 1 second. In total, the 50-second observation. So the details are here. So condition 1 is only observe a pupil oscillation in the 10 seconds. Condition 2 and 3, so blue light pulse for either eye. Condition 4 and 5, red light pulse either eye. So the pupil reaction was measured using apparatus Hitomir. It's like this. It's a goggle-style pupil observation system. So also observation was conducted in a dark room, and this Hitomir is only measuring system. The lighthouse added to this system. So blue light and red light, and so this wavelengths and luminance is mostly same level. So pupil size sampled in the 6 hertz. Using this equipment, pupil diameter was measured, and how to say, the eye blink usually frequently happened. These blink artifact manually compensated. So observed both eyes, either eye or mean of both eyes. So here is an example of PLR. Left figure shows normal control. So once restrict as 1 second light flash, and restoration phase is exist. Now for the right side figure shows Alzheimer patient. So once shrinked, but the restoration phase may be influenced by disease as a little bit abnormal. So, and we extract some features from PLR waveform. The definition and extraction procedure was developed in our previous study in 2018. So here, here is a waveform. We focus on restriction and restoration phase, and 10 variables are extracted. So, and we compared features between the both eye. So, however, only one RA, relative amplitude of meiosis is significantly different on only one conditions. On other features and all conditions, there are no significant difference. So then the little difference between the two eyes, the average of two eyes was used as a future. In order to reduce the number of futures, factor analysis was conducted. The three-factor scores extracted. So three factor. First factor is like this. So velocity, acceleration max, and so second factor is size and time at minimize, and factor 3 is time factors. The factor score compared across the so observation conditions, 4 conditions: blue light pulse or left pulse, red light pulse or left pulse. Here is the factor scores. So as you can see, the factor scores in typically factor 2 scores decrease in experimental order. So then the experimental order may affect the scores. So we compare the factor scores between the conditions. So in focus on the blue light, so there are significant difference in the factor 3 in the blue right and left conditions, and red light condition is sorry, it's only 3. Okay, how to say two conditions. So and we also compare the subject group, 3 subject group, and so there are no significant difference in futures and factor scores. We conduct examine these factors. Two-way ANOVA was conducted. So age group was significant. So we introduced logistic regression to classify the patients and normal control. And so initially, 10-dimensional future introduced like this. And so in this study, so we use AUC for the performance index. There are 10 variables introduced, 0.84, and to extract some major futures. So variable selection technique was conducted using stepwise method. The 5 variables in the blue condition was extracted, and age group. So once again, we set we use a 10-dimensional future. The selected 6 variables is factor 1 and factor 3 is blue light 2 conditions, and factor 2 was over blue light conditions and age group. So this selected variables shows dominance of PLR futures for blue light. So logistic regression calculate the possibility to the AD or MCI. So horizontal axis indicate MMSE and vertical axis indicate probability. So as I mentioned, here, less than 23 is possible is a AD group. Here is MCI and normal control. If we set to the threshold for 0.5, so they are patient, and they are here is normal control maybe. So we confirmed the age factor. So horizontal axis is age and vertical axis probability. So as you can see, probability is stand up over 70 years old. Anyway, so we introduce a threshold, and we classify to the 2 groups, AD or MCI and normal control. I mentioned that AUC is 0.85. 84, the ROC curve is like this. So we also confirm age factor again. So we compare the probability between the AD plus MCI and NC. So as you can see, the slope to the probability is totally different to the patients and normal control. So here is a discussion. So we compare the future and factor scores between the both sides. However, in most cases, not significant. So to detect AD and MCI participants, so as a early detection procedure, so MCI detection is very important, but so small numbers because MMSE scores is a too narrow range, so then they're not easy to detect. So PLR features for blue light pulse is significantly selected, or so regression functions. So here is a summary of my results. And so for future work, is redefinition is required for the PLR features and also compared to the other metrics like this. So finally, authors appreciate to the collaborators to conduct experiment at the hospital. So thank you for your kind attention. Thank you very much.
Thank you, Minoru. I received 2 questions from Dennis, but one regarding the limitations. You already said a bit about it in your discussion. And the other one was the paper addresses the contribution of the participants' age. But did you also check for the contribution of sex, since other research shows higher rates of dementia in women in late life?
Okay. So yes, you mentioned too many factors. So, but
did you also consider the sex, not just age, for your
okay,
to
yeah.
have
So,
a more...
so we should consider the sex, but we the how to say so there are no significant between the sex difference. I think I forgot so little bit so the values, but so not significant. But so age is significantly influenced because so how to say pupil dilations is so influenced by age. Well, generally, so pupil shrinking by age. So then the pupil oscillation or so dilations may be influenced. So that's a major factor, I think.
Yeah.
Okay.
Yeah, and he just then seems to refer to some research that shows that there are more women who have dementia in the older age.
Yeah, yeah. So you mean the sex difference on the dementia?
Yeah, he asked if you checked for the contribution of sex in your study.
Yeah, yeah. However, I explained to you. So we check sex difference. However, so we cannot detect significant difference between the sex difference. However, so as you can see, so the we gathered randomly the patients. As a result, the more patients include female patients are included. So that means how to interpret it, this phenomena we have to think about carefully. That's our future work.
Yeah. Would you consider your study representative?
What do you mean representative?
Do you think that your study is representative? So does it is it a balanced division between everything? Does it represent the population?
Yeah, to tell the truth, we cannot think about these factors because how to say patients gathering is a very serious problem. We ask hospital to conduct observation. Usually not easy to gather the patients. So then we usually test it only as 10 or 20 scale. So in this experiment, we get collaborator as hospital people, and we can get 100 participants. So, but not only as patients and family members we observed. So then we gathered 100 scale.
Yeah. Thank you.
Okay.
Are there any other questions? I see a hand from Franziska.
Thank you. My question is, I remember your last year's paper and presentation, and I am interested in the path you took between the old paper and the new one. Were there any learnings that you could take away?
Yeah, so last year, so we presented pupil response during how to say medical consultations. So this year, so very conventional experimental style is observed only as PLR. So the experimental style is totally different. So we hypothesized the how to say unsynchronization between the two pupil response when the patient in the patient. However, we tested the how to say synchronization. We cannot find out no significant difference. So this will be our future work. We try to check again the difference because we hypothesize here. So this how to say this experiment based on this how to say clinical situations. So retinal connections is maybe influenced by Alzheimer's disease or not. So we want to confirm, but so we cannot resolve this hypothesis. This will be future work. Okay.
Thank you.
Thank you very much.